Hola, ciao, konnichiwa, hello, um, hiya, howdy, partner, um, all of those things. Hi guys. So today I'm going to do a little video recording on the second part of chapter 16, the molecular basis of inheritance. All right, and we're still focusing on DNA. So let me share my iPad. My son was a pirate for Halloween, so there's been a lot of R jokes. R matey. R R R. Okay. Okie dokie. All right, so we left off last time with the structure of DNA, and I'm not going to draw it again, but I just want to do a couple reminders. Um, so remember that we have a 5' and a 3' for every piece of DNA. I usually draw an arrow on the 3 end because that's where new nucleotides are going to be added, right? So we would have 5' to 3' in this orientation. Those bonds that are holding together this backbone that I've drawn are sugar phosphate bonds, right? Otherwise called posh. Whoopsie. Phosphodiester bonds, right? For the backbones. And then towards the inside, we are putting our base pairs. And remember, anytime you have an adenine and a thymine, you're going to have 2 hydrogen bonds. If we had a cytosine and a guanine, we're going to have 3 hydrogen bonds, right? Remember which ones are purines. Purines, remember pure as gold, adenine and guanine. Yes, you have to know these names. And then the pyrimidines are cut the pie. So we have cytosine, uracil, and thymine, all right? And remember, uracil and thymine just depends on whether you're talking about DNA, thymine, uracil, RNA. Okay, and remember what this 5' means, right? It has to do with which carbon we are dealing with. So remember, we go clockwise from the O, 1, 2, 3, 4, 5'. If we're talking about the 5' end, we are talking about a phosphate up there. If we are talking about the 3' end, we would have in OH. And remember that OH is absolutely critical for the addition of new nucleotides. Okay, so we've talked about the DNA structure, and we kind of ended off with Watson and Crick. So in their 1953 paper, which is again posted online for you, it says this quote, which I love. It says, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." So you have to remember at this point, this is the first time we knew that DNA really was the genetic material. And for the first time, we knew the structure, which was a huge, probably the biggest breakthrough in science, possibly of our lifetime, of our generation of science. So for the first time, we could look at the instructions of how any organism is made, and then we start to figure out what that means, right? And that's what the next several decades we're up to. All right, so if you look at the parent molecule here, over here, if you look at the parent molecule, the mechanism of replication that he is alluding to is that these split, giving you 2 template strands, right? They both make a template for the other. Then they both duplicate or replicate via this one word that you're going to use often, and it's complementary base pairing, right? And this allows us to give you 2 exact copies, right? So again, if you split it up, now we have this A, it's going to be T, G, A, T, C. And we'll do the other side, A, C, T, G, right? Giving us these two now identical copies due to complementary base pairing, which is something that you're going to use on your test. That is a very important phrase, complementary base pairing. Just want to point out that the enzyme that does this DNA replication is called DNA polymerase because it polymerizes or adds nucleotides to make a polymer of DNA, right? So the DNA polymerase catalyzes, and again, it's an enzyme. Anything with ACE is usually an enzyme. So DNA polymerase is an enzyme that catalyzes the addition of a nucleoside triphosphate, all right? And I'm just gonna box that. So this is our nucleoside triphosphate. And that is what actually gets added when a DNA strand is growing. So a few important things. One, you have to have this 3' OH, all right? This 3' OH then basically attacks right here, giving you a pyrophosphate, which you don't have to know the name. You basically get two inorganic phosphates coming off, right? To add your phosphodiester bond, right? Which is the connection between your phosphate and your next nucleotide. So remember, every nucleotide, has a phosphate group, a 5-carbon sugar, and a nitrogenous base. And this here is the phosphodiester bond. So there was another experiment. I didn't include this in the hunt for the experiment, in hunt for the DNA, because this one's a little different. It's called Messelson and Stahl, and these guys proved how DNA was replicated was through a mechanism called, which they termed semi-conservative replication. All right. So before we talk about that, I really want to talk about down here, the three different models for DNA replication before they started. All right. The first was called a conservative model, in which case both, like a strand of DNA, let me see how to say that. So basically, where you have a strand of DNA, and when it was replicated, that stayed put, and it somehow read that, and you got two new strands, right? So that is a conservative, meaning that the original DNA is completely conserved, 100%. And then new DNA, as it's being made, is two new strands, all right? The second model is semi-conservative replication, which is how it actually works, where you have a strand of DNA that gets separated into two strands, and then the new DNA is read from those templates, right? So it's semi-conservative. So one strand is conserved from both when you make the new strands, right? And the third model is called dispersive. And this really was a model where you had your original DNA, and instead of it completely separating and having each one read, it had little pieces read, right? So for example, you'd have little pieces of old DNA, and you would have the new DNA kind of interspersed throughout that model, all right? So we now know it's conservative replication, and we're going to talk about why. And it's through this experiment called that was done by Messelson and Stahl. So the experiment is that they grew up bacteria. All right, so they're using bacteria here. They grew up for several generations, and they used then several generations in a media, or in a kind of the food for the bacteria, that contained a heavy isotope of nitrogen. All right. So what they did is they grew it in nitrogen. Why would N15 matter? Because DNA has nitrogenous bases, right? So it would have radioactive or N15 incorporated into its original strands, right? So if we had N15 here in black, then every piece of DNA would have DNA that had N15 inside of it, right? As it was growing in that original bacteria. Then they did something really clever. They then transferred that bacteria to a media that had only N14, right? Which is a lighter isotope. And then they took DNA samples that had replicated, and they looked at what happened over generations, all right? So for example, if we have N15 here in black, and then we're gonna start putting it in N14, that means that as it replicates, we're gonna have half of the strand, one half strand N14 and half strand N15, right? So the black is N14, the black is N15, the red is N14. So what you see now is that in generation 1, we have 100% N15. In generation 2, we have half N14, half N15. So they're all hybrids, right? And then they looked later and later. And they did this through a centrifugation where you can literally separate the DNA that is more dense, meaning N15, versus lighter or less dense N14. All right. So let's, Mr. Anderson also does a nice job of explaining muscle and stall if you want more information. But here I want to show you what the data actually can look like. All right. So here we're talking about generation number 1. All right. So in the first generation, all of it is N15, right? All of it is N15. That's originally how it was grown. All right. Then, so that was the first, that was at time 0. At generation 1 here, we can see obviously that we are only dealing with N15. In generation 2, you can see that there is 50% of the DNA is N14 and 50% is N15. So what that means, again, is that the DNA separated and we had 100% of the DNA, 100% of the DNA is in this hybrid form, right? Giving us 50% N14, 50% N15. Then they looked at generation 3. And I highly recommend that you draw these out if there's a question on this on the test. So in generation 3, spread out, these guys separated, right? So we had those separated and these separated, and they're still growing only in N14. So now every new strand is N14. And what you'll see now is that 75% is N14, right? And you can see that 75% versus 25. And I recommend that you do it again with generation 4 and convince yourself. So again, each of these are going to separate out. So we have the blacks separated, and then we have the red. So we have one there. We have 2 of those. We have 1 of those, and we have 2 of those, right? So we have 1, 2, 3, 4, 5, 6, 7. There should be 8 strands of DNA. We're going to end up with 16. Every strand is going to get a red, right? So sorry, my DNA is getting a little messy here, right? And now what we can see is that we are going to have, a different amounts, but again, higher in the N14. So hopefully that makes sense. And here's a type of questions, just so you know what's happening. So these are the kinds of questions I might ask. And then late 1950s, Messelson and Stahl grew bacteria in a medium containing heavy nitrogen, and then transferred them to a media containing N14. Which of the results in the figure would you expected after 1 round of replication in the presence of N14, right? A, B, C, or D after 1 round. So again, don't pretend that you can, or some people can just do this in the head. I always have to draw it out. So after 1 round, we are going to see the parent strand separate, and then we're going to get new DNA. So we would expect to see all at least 50%, right? So we'd have, so here's N15, here's the N14, here's the hybrid. So we would expect what? All of it to be in the hybrid. That makes sense. So at the beginning, all contains 15. After the first round, all the DNA will have 1/2 N and 1/2 N14, 1/2 N15, right? So after 1 round of replication, we would expect all of them to be in the hybrid. So that's a type of experiment that you should be able to answer. All right, I just want to now go back to the sugars, which we've kind of gone over before. So we'll do this quickly. Remember that the carbon numbering is absolutely essential, that you understand that you start at the 0 and you go clockwise. 1 prime, oops, 2 prime, 3 prime, 4 prime, and 5 prime. Remember, the 5 prime carbon is out of that ring, right? And nucleotides can only be added to the 3 prime end, and that 3 prime must have an OH attached to it. Therefore, replication can only happen on the 3 prime ends. And we call that 5 to 3 prime, because as we're going to add, we're going to lengthen this part. All right. And again, how they get added as a trinucleoside or nucleoside triphosphate. But again, these 2 inorganic phosphates come off. Okay. A couple things to think about is that DNA does not exist naked in the cell, right? Rather, DNA exists on chromosomes, right? If you're a prokaryotic cell, you have a double-stranded circle. If you are a eukaryotic cell, you have individual linear chromosomes. And a chromosome, what chromosome really is that it's DNA and proteins, right? And in particular, the proteins are called histones, right? And these histone proteins, which are down here kind of in this purple blob, the DNA wraps around an octet of histones. So you get 8 of these little histones coming together. And 1, 2, 3, 4, 5, 6, 7, 8. Yep, those 8 histones are going to come together. They form these little kind of octets, and the DNA wraps around them. All right. And you can see the DNA wraps around the histones. Each kind of histone blob is called a nucleosome. And then it coils and coils, and it's called a supercoil. And that's how a huge amount of DNA gets into a very, very small nucleus. When DNA is not dividing, it's wrapped around these histones, right? When DNA is dividing, kind of some of these histones, they have to loosen up so you can expose the DNA, right? And same as we're going to look, remember that we always go from DNA to RNA to protein, right? And what we're going to see is that this process here, when we go from RNA, when we go this process, DNA to RNA, we're also going to have to loosen up those histones to expose the DNA when we kind of transcribe them, which we'll get to later. All right, so DNA always replicates at a very specific part of the cell cycle. And what I mean by the cell cycle is that every cell has a kind of characteristic cell cycle for that cell. And it always starts in this kind of way, is that you have a growth phase, right? So the cell kind of starts growing. Then you have an S phase, right? DNA synthesis. The S stands for synthesis, right? So that is where you get DNA replicating. Then you get growth and preparation for mitosis. And mitosis, as we're going to learn in the next section, really is nuclear division, right? That's when the nucleus of the cell is going to divide, when the cells split, right? So you get growth, preparation for that, and then you're going to get mitosis or cell division, right? So what we're going to see is that one cell with one copy of DNA is going to divide into 2 cells, each with 1 copy of the DNA. Sorry, we'll talk about that more when we get into mitosis. So the point is that these phases, G1, S, and G2, all stand for interphase. And a cell spends 90% of its time in interphase. It spends a very, very small part of its life cycle in mitosis, or when the cells actually split, all right? DNA is synthesized in S phase, which is part of interphase, right? This is where chromosomes get and then And then during mitosis is where you are going to separate the daughter cells. So just as a just to go over that again. So when we go through this part where we are making new copies, that is going to be S phase, right? When cells physically divide, right? We are going to see that when the cell actually splits into two, that is going to be for in mitosis. And we'll talk more about that when we get there. So DNA replication, which is really what we're focusing on, always begins at sites called origins of replication, all right? Sometimes those are called ORI, right? And I'll probably say that just because I'm lazy. ORI, or origins of replication, all right? And they're dictated by certain sequences, specific sequences or nucleotides within the DNA sequence. And the DNA starts separating at that point to create what are called replication bubbles. All right, so let's look over here at figure 16.12 first. Here we're talking about the origin of an E. coli cell. So E. coli is bacteria, right? Remember, they have a double-stranded circular piece of DNA. They have a single origin of replication, right? Little ORI, a single origin of replication in prokaryotic cells. So what happens is the DNA starts to separate there, right? And as it separates, you create two sides. Well, let me start. You create what's called a replication bubble, right? And you have two sides of that replication bubble, right? So I'm going to do it kind of over here. So as it opens up, you're going to get one replication bubble, but each replication bubble has two replication forks. And all it is a fork in the road. So you go from, two strands and they kind of separate. So two replication forks, one replication bubble. And as you trace that along, as that bubble gets bigger and bigger and bigger, what eventually you will get is two daughter DNA, right? Two new daughter DNA molecules, both with identical DNA. So circular bacterial chromosomes have one single origin of replication. However, eukaryotic cells over here, eukaryotic cells, are much more complicated. They have a lot longer DNA sequences to deal with. Even though they're linear, they're way longer, more complicated. So we have what are many origins of replication. So we have one here, and one here, right? Each of those origins of replication are going to start by opening the DNA, separating those two parental strands to create replication bubbles, right? Every time you get a replication bubble, you're going to again have two replication forks. And when we draw this out, hopefully it'll be much more clear, all right? And eventually, as you make that new DNA, the daughter DNA here is in that light blue, right? That's the new DNA created through complementary base pairing. And then eventually you get two daughter strands with identical pieces of DNA. So eukaryotic linear chromosomes have many origins of replication, bacteria only one. So here are the important proteins that you're going to have to know about that are involved in DNA replication, right? We have DNA helicase, right? So helicase is shown here. Oh, hold on, let me see if I can color code it because I can. So DNA helicase here is shown in this little green molecule. What it does is it opens up the DNA, starts at that origin of replication, and it literally pulls the DNA apart and separates those two strands. So what kind of bonds is it breaking? It's breaking the hydrogen bonds between those nitrogenous bases, right? As it pulls it apart, I don't know if you've ever taken a piece of yarn that have multiple strings and you pull them apart, the ends get super tangly, right? So there's a molecule that I don't really care that you know about, but I'm going to mention it because it's rad, which is called topoisomerase. So topoisomerase basically prevents that crazy winding as helicase as helicase opens the parental strands, topoisomerase kind of is downstream of it, meaning it's down from where helicase is heading, because helicase is heading in this direction as it unwinds it. And the topoisomerase prevents the DNA from getting super tangled. All right. Another one that I'm not going to make you know on a test is called a single-stranded binding protein. And this just stabilizes the DNA as it unwound. So helicase opens it up, single-stranded binding proteins bind that single strand, good name, and prevents anything from crazy happening. All right, but the ones you do have to know about. So primase. Okay, so primase is this nice little pink one. And it synthesizes what are called RNA primers. So it uses the DNA parental strand, and it uses, wait for it, complementary base pairing to create what is an RNA primer, right? Complementary base pairing, all right? And that's because, again, in order to get DNA polymerase to work, you need what? You need a 3' OH, right? And that RNA primer provides that right there. Okay. Then we have what is called DNA polymerase. All right. That's not actually shown in this picture. The DNA polymerase is a big old molecule that is going to make all the new DNA by adding it to the 3' end. It reads the template strand, adds a complementary base, right? And then last but not least, we have DNA ligase, which is also not shown here, which joins the sugar phosphate backbone. All right, and I'll show you what that does when we draw this out. But DNA ligase is going to be really important for, in particular, that sugar phosphate backbone. All right, so there's lots of rules of DNA replication. I talk about it in a very specific way, but the way I talk about it doesn't always work for every student. So I like to give these rules in case other people like to hear them this way. So DNA is always read in a 3' to 5' direction. So what I mean by that is as DNA polymerase sits on the DNA, it is reading the template strand 3' to 5', right? So reads the template strand in a 3' to 5'. The DNA is always synthesized 5' to 3'. So that's the new strand, right? So as you can see, polymerase would be moving in this direction, reading that template strand. However, the new strand is going from 5' to 3'. Helicase will sit right at that opening, right where the two strands are coming together, and it's going to unwind the DNA. Primase is going to be, it would be right here, for example, right before the DNA is being made. Primase is going to start, here, I'm going to actually do this one, start the synthesis of DNA, right? So it adds an RNA primer from which DNA pol can bind. It's going to read the template strand, and it's going to create a new strand, always on that 3' OH. The DNA polymerase is going to be the one that reads the strand, adds complementary base, reads the template, adds complementary base. And then we're going to see DNA ligase going in afterwards and super gluing these pieces together on the new strand. All right, so I've already filmed drawing out this synthesis together. So this is another one that would be really useful to watch. Okay, so some important proteins during replication. We've already talked about these. So we have primase that is important for the synthesis of RNA. We have DNA polymerase, which is going to be important for elongating that new strand. And DNA polymerase will also, when RNA primers have to get removed, and they get removed, so these are eventually removed by what's called an exonuclease. And then RNA primer will come, DNA polymerase will go back in and fill in those the spaces that RNA primers were there with DNA, right? And what you will see I'm going to go back to the strand. So what you're going to see is that as the DNA is replicating, so here's one replication fork, right? Remember, in a replication bubble, you have two forks. We're only looking at one of them here. And what you're going to see is that one strand can just continue to be made into that fork. That is called the continuous or leading strand. The other strand, due to that DNA must be made in this 5' to 3' orientation, it's called the discontinuous or lagging strand. All right. So if we go here, the synthesis of the leading strand is pretty straightforward. The synthesis of the lagging strand leads you to have many little segments of new DNA, and those are called Okazaki fragments, which then get joined back together with their DNA backbone by DNA ligase, right? And again, as we draw this out, hopefully that'll make more sense. All right, so a couple things. So there are errors when you do DNA replication. About 1 in, I don't think that 110, I think it's 1 in about a million nucleotides, there's an error. Sorry, I'll check that number for you. But during synthesis, oh, maybe that's correct. Okay, so 1 in about 1,000 nucleotides is an error. And then, sorry, let me rephrase. Okay, so when you look at DNA, there are some errors, all right? But during synthesis, we're gonna see that there are less errors than what we actually see. I'm not saying this very well. Don't hate. DNA polymerase can make errors, but the whole point is that it can fix themselves, all right? So there is what we call proofreading and repairing of the DNA, all right? So let's talk first in this one here, where we see that DNA polymerase here in green, this green molecule, this is your DNA pol. All right. And it read a T on the an A on the parental strand or the template, and it accidentally put in a C, which is like major error. So what DNA polymerase can do is it sees that error. It feels that for example, the width of the DNA is not quite right? It can sense that, and it'll go back out. It will take out that mistake and it'll put in a new one. So this is called mismatch repair, where it can proofread and fix an error, all right? Sometimes we can have, after DNA polymerase has already done it, so here is a new piece of DNA, there was obviously an error right here that DNA polymerase didn't catch during the proofreading. So what happens is that you get what's called a nuclease enzyme. I usually call those exonucleases because they take DNA out. So it uses little scissors. Those are my little scissors. And it cuts out that piece of DNA that is wrong, and DNA polymerase can go back and fill it in. Very similar to what happens with RNA primers. And then DNA ligase can go back in and make sure that the phosphate sugar backbone are all glued together. All right, so at the very, very ends of chromosomes, so if we look at a linear chromosome, we have these kind of very special ends, all right? And those are called telomeres, all right? And they are the aglet of chromosomes. I don't know, I hear your generation, there's a song about aglets. We'll listen to it in class maybe. So the aglets are like the thing that are on the end of your shoelace that keeps the shoelace from unraveling, right? And telomeres are on the ends of chromosomes and they help them from unraveling. So telomeres are a special repetitive DNA nucleotide sequence at the ends of these eukaryotic linear chromosomes, and they protect the DNA or the genes from being kind of eaten down after many rounds of replication. And I'm not going to go into the details of telomeres, but there's this awesome enzyme called telomerase, which is used. It's an enzyme that catalyzes the lengthening of telomeres to restore their length every single round of replication. And there's this badass lady that I used to know at UCSF named Elizabeth Blackburn, who discovered telomerase. And she got the Nobel Prize back in the day. I forget what year. But she's amazing. But telomerase is very, very cool. You should check out this diagram and see if you can explain why telomerase would be important. All right. And again, lots of practice questions at the end of the PowerPoint that you should do. All right. Peace. Bye. Ciao.